So Sarah, I'm glad I bumped into you because Rob's been talking about these phospholipids and how they can build up into like a membrane and how those membranes are fluid. And honestly, I don't understand a word of it. So I know like a phospholipid looks something like this, but I don't really know how to make heads or tails of this particular chemical structure, what I should be interpreting from it.
Okay, I totally understand your confusion, Marty. This molecule looks a little bit weird with all of the atoms, but basically what we can do is separate it into, exactly as you said, heads and tails. So all we're really looking at is, do you see that there's kind of this cluster of molecules up here, of atoms up here? This is just the head.
Okay.
So this is one phospholipid, there's the head, and then down here there's these two long hydrocarbon chains. These are the tails. So it's that simple, but we use a lot of terms to describe the differences between the head and the tails because that's pretty important to the function of the molecule.
Okay.
I don't know if you remember Rob saying any of these terms.
Yeah, I think I remember him saying that like the head was polar, which seems to make sense given the atoms that are present there. And so if it's polar, I imagine it's hydrophilic. And these long hydrocarbon tails, I guess those are hydrophobic because I don't see any polar atoms there.
That's exactly right. So all we're looking at is the head, it's polar, it's hydrophilic, exactly as you said. And because it's hydrophilic, that means it's water-loving. So the head of a phospholipid is going to always want to interact with water. It's going to want to be next to water. And then the tails are nonpolar, exactly the opposite of the head in every way. They're hydrophobic, and this means that they're water-fearing.
Got it.
But we can take all of these traits and this entire structure, and we can kind of simplify it into something that looks sort of like this. You can see a head and two tails.
Yeah, okay, so I see what you're saying. So this particular shape right here, the two tails are kind of clearly these two hydrocarbons that are nonpolar, so these are the hydrophobic tails. And I guess from that, I'm assuming that this circly thing up here, that's the polar head, that's the hydrophilic head.
Yeah, exactly. So these are just two different ways of representing the same thing, which is a phospholipid.
Okay, so a phospholipid is a molecule that has a hydrophilic head and two hydrophobic tails.
Yes.
So I still have no idea how that actually helps me understand what a membrane is.
Okay. I see the confusion because, you know, when we have a membrane, we're going to have to put lots of different phospholipids together. But I think the simplest way of understanding how to make a membrane is to look at what happens when you actually add water to the mix.
Got it.
Because again, we have these water-loving head, water-fearing tails. So what would happen if you took a glass of water? You have one right here.
I have to stay hydrated.
Yeah. So let's just draw your glass of water. What would happen if you took our phospholipid and just dropped it in to the glass of water?
Okay, I see what you're getting at. So if we had to interact with water, I'd probably imagine that if I drop this phospholipid in here, that the polar head would interact favorably with the water that's in the cup, and the hydrophobic tails would have to orient themselves in some way that they're pointing away from the water.
Yeah, that's exactly what would happen. In fact, let's go ahead and why don't you just drop a bunch more into the glass of water and
see what
happens there.
Okay, this is cool. So I assume that they'd all be doing the same thing, right? So we'd have all of these different phospholipids that are orienting their heads towards the water and orienting their tails away from the water.
Yeah.
Why'd you give me all these ones with different colors, by the way?
Oh, okay. I guess that is confusing because in the video they're different colors.
Right.
But we don't need to dwell on it too much, but there actually are different types of phospholipids in the cell. Well, the one we looked at originally, this one is called phosphatidylcholine, and it's just one type of phospholipid. There's other types too, and they mostly look the same. You can see the tails look the same, but they have different names. Here's phosphatidylserine, and so I don't know what differences do you notice?
So it looks like the hydrophobic tails are basically the same for all of these, but it looks like the head groups are different. So this one has like a sugar attached to the phosphate. This one has like a nitrogen and two carbons attached to the phosphate group. This one just has slightly different nitrogen arrangement with the carbons attached to the phosphate group.
Yeah, exactly. So the head regions are going to be slightly different chemically in each case, and so we represent these as different colors, but ultimately all of the phospholipids will behave the same way in water, and they'll all kind of be smushed together in a membrane. So that's the different colors. It's just slightly different head groups and different types of phospholipids.
Okay, so I'm loving this. So we have like our phospholipid layer that's existing on top of our water, but of course, like a cell is mostly water too, right? So how do the phospholipids arrange in like a sea of water when most of the inside of a cell is also water?
So that's a good question. Let's draw this out. So we have, let's say we have part of a membrane here, which we just drew this. We just looked at how this would form if you took a bunch of phospholipids and put them in water. You get something kind of like this where they're all aligned.
Sure, yeah.
And
all the hydrophobic tails are pointing in one direction, all the hydrophilic heads are pointing in another direction.
Exactly. In the cell membrane, we have a phospholipid bilayer. So we'll take a second one of these and line it up. And what do you notice here?
I see. So this like in this kind of a bilayer, so like this is one layer, this is another layer. And in this case, you could have water molecules on both sides of the membrane, but you'd have like no water on the inside of this region where the hydrophobic tails are.
Exactly. So that's the exact case we have with the cell, because the cell will have water on the outside and it will have water on the inside. So this way we keep the phospholipids entirely happy. The polar head groups are very happy because they get to interact with water on both sides, and then the tails are shielded from water. So this region in the middle, inside the phospholipid bilayer, will have no water.
So this is a phospholipid bilayer, and this is what like our cell membranes look like.
Exactly.
So then Rob talked about how these things are like fluid, and I don't know. So I'm just like looking at this now, and I'm thinking when he says fluid, that means because like the phospholipids aren't attached to one another, so they can kind of move around. So what happens if I like they can do that?
Whoa. Okay, they can not do that. They can do a lot of things, and you're right. They are really mobile, and we call the bilayer fluid. But you can't actually have a phospholipid jump from one leaflet of the bilayer to another. But what you can do is you can kind of rearrange them relative to each other. So they'll shift around in position, and the cell, when we're looking at the cell, it's not two-dimensional like this. We're looking at a sort of a spherical shape, so you can imagine the phospholipids rearranging themselves within the spherical surface of the cell.